Have you ever wondered how DNA gets a trait, like, for example, the eye color? Yes, your DNA has the genetic information that codes for the color of your eye. The eye color is based on the pigment that is inside the eyes. But in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So, what we're going to talk about is how your DNA can lead to the making of protein. This process is called protein synthesis. Again, synthesis is simply means to make something. So, protein synthesis means to make protein. Proteins are so essential as these function almost everything inside the body. It helps in the buildup of muscles, the enzymes we use for biomolecule processes, and what is so cool is that, unlike the carbohydrates which we cannot make inside the body, you are making proteins right now as you sit and watch this discussion. It's happening in your cells. It's happening in the nucleus. Some DNA are non-coding DNA. Some DNA makes up genes that are not activated, but for now we will talk about genes that are coding for active proteins. How are we gonna get the information from these genes out of the nucleus so that cell can start producing the proteins that needs to make? In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has C in it and translation has an L in it. I remember as C comes before L in the alphabet, that helps me remember that transcription comes before translation. In your cells, the DNA is in the nucleus. So therefore, we're doing transcription in the nucleus. Transcription is where we're going to transcribe the DNA into a message. It is by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA. In this step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA or messenger RNA. mRNA consists of a messenger made of RNA that has been based on the DNA. To do this, RNA polymerase, with the help of transcription factors, binds to a specific sequence within the gene which is called the promoters. These promoters prise the two strands apart. One of the strand will serve as the template strand or antisense strand, meaning it will be used to generate the mRNA, and the other is the non-template strand or sense strand. RNA polymerase doesn't need a primer. It simply initiates the mRNA synthesis at the start codon and then moves downstream along the gene in a process called elongation. Synthesizing the mRNA as it goes, reading the anti-strand from 3' to 5' and generating mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which we call ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from gene and the DNA returns to its original state. But we now have the produced mRNA. This carries with the information encoded in the gene. As you have to take note that this mRNA is not ready to go right away. There is usually a significant amount of mRNA editing that occurs. This is what we call post-transcription, and after a few quick modifications during RNA processing, it will leave the nucleus where all the genetic material or chromatin is, and move into cytoplasm where it will find a ribosome. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. And it is in the ribosomes that we make protein. And that's easy one to remember because the rRNA, in which the R stands for ribosomal RNA. The ribosomes is going to build our protein in the next step called translation. During translation, the mRNA acts as a code for a specific protein. This happened because each set of three bases on the mRNA, which we call codons, will code for a specific anti-codon, which will be carried by a specific tRNA. Since there are four bases and each codon has three letters, 4 to the power of 3 gives us 64 different possible codons. So how to identify your genetic code? Using this table, you look for first letter of the code. Again, the code comprising three letters. The first letter are located at the leftmost portion of the table. Let's have the example G, C, A. Since the first letter is G, we look first on where all the first letter G is. Therefore, all of the genetic code with G as their first letter is located here. Next letter for our example is C. Second letters are located at topmost of our table. Therefore, the genetic code based on our example is alanine. Notice that there is some redundancy with multiple codons resulting in the same amino acid, but there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some codons are special. AUG is the start codon which initiate translation by coding for methionine, and these three are stop codons. These are those that terminate the translation. In the cytoplasm, you have these tRNA or transfer RNA molecules anticodon available. It's easy to remember as the shape of the RNA is like a T letter. Each different tRNA is covalently linked to a particular amino acid. The arrangement of nucleotides into this codon is called the reading frame. The reading frame will be what the tRNA paired with. In the translation, we have to take note of the four basic steps: activation, initiation, elongation, and termination. First is for activation. The RNA will be activated once there is an amino acid attached to it, covalently bonded by ester linkage. Once activated, it will now go to the next step, in which is the initiation. Translation occurs inside the ribosome. Ribosome comprising two parts, the large ribosomal subunit and the small ribosomal subunit. The small ribosomal subunit binds to an mRNA and an initiator tRNA adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. After this, elongation process takes place. The tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid which become covalently bound to the methionine of the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifter over making room for the next tRNA. The new amino acid links to the first two, and this process continues all the way down the mRNA. As the RNA enters and exits the ribosome in a sequence that dictated by the codons on the mRNA, a polypeptide chain will grow. This continues until a stop codon is reached. At this point, the completed polypeptide will swim away, most likely entering one of the cell organelles for folding and further modification. In summary, in this two-step process, DNA is transcribed into mRNA, and then this mRNA is translated into tRNA, and then this tRNA is translated into a protein. all simply by obeying base pairing that occurs in the nucleic acid, and since every gene codes for a specific protein, the proteins make up most of what you are. From your muscle tissue and organ tissue to all of the receptor and enzyme, this is how DNA carries the code for a living organism. Some antibiotics function by targeting the processes we have discussed. We call the group of such antibiotics as protein synthesis inhibitors. In general, protein synthesis inhibitors work at different stages of bacterial mRNA translation into proteins, like initiation, elongation, including amino acyl tRNA entry, peptidyl transfer, and bacterial translocation, and termination. Erythromycin, for example, the drug of choice of Legionella pneumonia and effective in the treatment of chlamydia infection, specifically binds to a large ribosomal subunit of the bacteria, thus inhibiting the exit of the RNA. In this manner, since the RNA can no longer exit, there is no new room for new tRNA in the large subunit. Thus, the growing of peptide chain will not be possible and protein cannot be made. Terramycin, on the other hand, that is a drug of choice for atypical pneumonia or pneumonia that has a milder symptoms that is usually seen on the infant and children, specifically blocks the A site location on the ribosome, preventing the attachment of amino acid carrying tRNA. Streptomycin, that was considered as the first drug to be successfully used against tuberculosis, or commonly known as TB, is the opposite of erythromycin. Instead of the large subunit, streptomycin focuses on binding on the small subunit. Once binded, the shape of small subunit will be changed, causing a misreading of mRNA information. Neomycin has similar function with streptomycin. And lastly, the drug of choice for treating infection caused by Salmonella typhi. The Salmonella typhi is the bacteria that we could get from raw poultry such as eggs. This is the reason why you have to cook your eggs kahit malasado, basta you could subject it to the heat to kill the bacteria present there. Chloramphenicol binds to ribosomes entering to the peptide bonds of one amino acid to the other.